Hi, welcome back. This is our Extensions in Mendelian Genetics lecture. So far in this unit, we have been talking about genetic traits. In fact, our big overarching question has been, how do I get my traits? And up until now, we've been working in Chapter 3, and we've been talking about Mendelian genetics, the stuff that Mendel figured out. But now we're going to move on to what Mendel didn't know, and that's in Chapter 4. At the end of today's lesson, you will hopefully be really clear on learning goal number 8, which means you can show how extensions and Mendelian genetics like codominance, incomplete dominance, and multiple alleles all can change our predicted offspring ratios. So again, this is using Chapter 4 in our textbook. Let's start by talking about alleles. As you remember, different forms of a gene are called alleles, and Mendel knew about those. But there's some ways we can work with alleles that he didn't know about. We know that mutations are the source of new alleles, and we call something a wild-type allele when it's the allele that we see the most in a population. For example, when you look around the room, most of the room has 5 fingers. That means you have 2 copies of a recessive allele for 5 fingers. 6 fingers is the dominant trait, a dominant allele. However, it is not the most common in a population. So, in this case, the recessive allele is the wild type, having 5 fingers. There's also something called a null allele. Sometimes the mutation stops something from functioning, and we call that a loss of function mutation. And so if we've got our wild type allele that's functioning normally, and then there's this different mutation that stops it from working, we're going to call that a null allele. Now, what Mendel didn't know is that more than one gene could work together to code for one trait. He also didn't know, he thought you always had 2 different factors for one gene, or 2 different potential alleles. We now know that you can have more than 2 alleles for one particular trait, or that can work for a certain gene. So we're going to talk about that today. Now, when we're talking about these extensions on Mendelian genetics, different symbols become important. Now, as you know, these symbols that we use are just a human construct. We made them up. You can use different letters and different symbols like variables. But it's important to know certain conventions are used at different times. And for example, so far we've been using capital letters to represent dominant alleles, but you could also use a superscript plus sign to represent a dominant allele. In that situation, a symbol without the plus sign would be the recessive allele. And we've been using so far a lowercase letter to represent a recessive allele. Anytime we don't have any dominance, which we're going to see coming up here in our incomplete dominance example, we are going to use our capital letters with different letters as superscripts to demonstrate that there is no dominance happening here in these different alleles. Now here's our first example. We're talking about incomplete dominance, where neither allele dominates the other. So what you'll notice sometimes is the offspring for that cross might end up looking like an intermediate between the 2. That's not a necessity. Really, what we would expect to see in incomplete dominance is a totally different phenotype for the heterozygote, something different from either parent. And when we look at that and we see that, that's usually incomplete dominance. And I want you to take a second and compare the phenotype ratio that you see up on this screen to maybe other phenotype ratios you've seen in the past. And turn to your partner and try to figure out what you notice there. What I'm hoping that you noticed down here is we see this very familiar 1 to 2 to 1 ratio, or 1/4 red, 2/4 pink, and 1/4 white. We're used to seeing that 1 to 2 to 1 ratio in genotype ratios, but not usually phenotype ratios. Because in phenotype ratios, usually the dominant homozygote and the heterozygote look the same. And so, as a result, they get clumped together in the phenotype ratio. So maybe we would normally see this as a 3-to-1. If this was regular Mendelian genetics, we'd expect to see 3 red to 1 white. But instead, we get 1 red, 2 pink, and 4 white. So this helps us see that this is not standard Mendelian genetics here. Now, in this case, it's most likely that the red flower is the wild-type allele. And somewhere along the line, there was a loss of function mutation that caused a white flower to happen, so that instead of making those red proteins, no proteins were made. And as a result, if that's happening, what would we call the white color allele? Think about it and try to write down the answer to that yourself. Now turn to your partner and see if you guys can share your answers. What I'm hoping you came up with was a null allele, because it was a loss of function. So again, loss of function usually refers to a null allele. So what we expect may have happened here with these flowers is you have your red flower, which has 2 functioning alleles that both can make that red protein. Or you have your white flower, which has no functioning alleles that can make the red protein. But when you have one of each alleles, one allele can make the red protein and the other can't, so we don't get as much red color. It ends up being pink. Another example for incomplete dominance is actually Tay-Sachs disease. And the thing about Tay-Sachs disease is you probably learned in school that you didn't probably learn that was incomplete dominant trait. You probably learned earlier, like, okay, if it was recessive, that if each parent had 1 copy of the gene, because it only showed up when they had 2 copies of the allele. Well, see, the way Tay-Sachs works is a good gene for Tay-Sachs makes a protein called hexa. And if you have hexa, it breaks down waste products in your neurons, in your brain cells, or in other cells. But if you don't have a good Tay-Sachs gene, or a good gene for that hexa protein, then you can't make the stuff that breaks down those waste products. And so those waste products build up all over your cells. And as a result, you get damage to your liver, eyes, and brain. So this is a very sad disease, and children die very young from this disease. Now, the weird thing about Tay-Sachs is it only shows up, it only presents itself as a disease when you have 2 copies of the allele. So we originally called it recessive. But it's a situation where this codominance doesn't appear in the heterozygous phenotype. So our homozygous recessive individuals are severely affected. They divide between 1 and 3 years. Our heterozygous individuals only have 1 copy of this mutant gene, so they only have 50% of the enzyme activity. So one of their alleles is working just fine in making the enzyme. And this is what's called the threshold effect, where 50% of this hexa protein is enough for you to function. So as a result, these individuals function normally. They never even know that they are carriers, that they hold 1 copy of this bad allele. And so a threshold effect happens whenever you have normal phenotype expression in the heterozygote, because you've made it to a certain level. Usually 50% or less of the gene product is attained. Take a second and answer this question. If Tay-Sachs disease is an incomplete dominant trait, why don't heterozygotes show the symptoms? Answer that on your own. You can go ahead and pause the screen. Okay, so now that you have written about the threshold effect and reflected on that a little bit, let's move on to codominance. It's very similar to incomplete dominance, because, again, it's not like one allele dominates over the other. But the difference with codominance is both traits are shown at the exact same time. And our classic example of codominance is roan cattle. If you have 2 copies of this B allele, you're going to have a black cow. If you have 2 copies of this white allele, you're going to have a white cow. But when you have one of each, you're going to see a roan cattle, which is pictured over here, some black spots and some white spots. You'll see both of them. Now what we're going to do is we're going to try to do an example with this roan cattle. On your own, I want you to cross a roan with a white. And I want you to figure out what the expected phenotype ratio is. And then I want you to think about this. Why are the phenotype ratios and genotype ratios, why do they end up the way that they do for roan cattle? Go ahead and try that on your own and pause me. So at this point, I'm expecting you went ahead and crossed your roan with a white. And you went ahead, and what it ended up looking like was this: 0 black, 2 roan, and 2 white for a phenotype ratio. The thing is, that would be your genotype ratio too. Because you don't get, no matter what, this parent is going to give a CW. So you're not going to get any CBs. And some of yours will get the CB with the CW, and those will be your 2 roans. Some of them will get a CW from this parent, and of course they'll get a CW from this parent, and those will be your 2 white. So again, we have a situation where the phenotype and the genotype ratios for codominant traits are the same. And I'm hoping you thought about that and realized the reason they're the same is because the heterozygote has its own unique phenotype. This is where incomplete dominance and codominance are similar. Both of them have a unique heterozygote that looks different from either one. But codominance, you get to see both traits at the same time. Now another example of codominance is the MN blood group. I'm sure you've learned about MN blood groups when, I'm sure you've learned about the ABO blood groups before. But MN is just another kind of protein that shows up on the outside of your blood cells, and they're little tags that say this is me, and those tags, if someone has a different blood type from you, are recognized and attacked by immune systems. So MN blood group is also a codominant trait. So individuals who with an M blood type have just the M tags on the outside of their cells. Individuals with the N blood type have just N tags, but MN blood types have both M tags and N tags. So go ahead, and I want you to try that problem for me. What is the probability that 2 parents with M and MN blood types will make an M blood type child? Give that a shot and pause me till you're done. So at this point, I'm hoping you've already done example 2 with the MN blood group for codominance. So I asked what the probability that 2 parents with M and MN blood types would produce an M blood type child. So this is what your cross would look like. The genotype for M is LM, and the genotype for MN is LM. So no matter what, we know that this parent is going to give an M. But this parent could either give an M or an N. So it ends up being a 50% chance of having an M blood type child. Now, multiple alleles is a situation where, again, Mendel didn't know about it. And again, a classic example of multiple alleles is A, B, and O blood type. So anytime we have more than 3 alleles in a population that are available for a certain gene, but again, each individual only gets 2, but there's 3 or more available, we're going to call that multiple alleles. So our best example for this is the ABO blood group. So we know that these are all examples of, there's all these different alleles. There's the IA, the IB, and the little I, where IA and IB are codominant to each other and little I is recessive to either. And these work together to produce the different blood types A, B, AB, and O. Now, at this point in time, you are going to go ahead and watch somebody else's podcast about blood types and Rh factors. And that may help you, especially if you've never learned anything about these before. And after you're done with that, I want you to answer the big ideas questions on the bottom of page 2 and on page 3. And when you finish that, I'd like you to go ahead and read pages 71 to 72 in your textbook. you're going to learn about something really interesting called the Bombay phenotype that also has to do with blood. The thing is about your textbook is I don't really like the diagram in there. So I want you to answer the 2 questions that I provided on your big ideas, but I also want you to take their diagram about the Bombay phenotype and I want you to make a better one. That is your homework. I hope this helped, and have a great day.